Welcome back. We're going to go on to developmental modes. I'm going to bop back to the last screen just for a second that we just left, or I just left. I had asked you before I signed off whether pseudocoelms are analogous or homologous. Do these two taxa, we'll just call them taxa R and taxa N, do these two taxa have pseudocoelms because they have a common ancestor that developed a pseudocoelm? No, they're just super separated on the tree. This guy doesn't. In fact, this guy has a true coelom. I guess this guy probably does too. These all have true coeloms. This one does too. This is an analogous characteristic. These two taxa evolved pseudocoela separately, but because they live under similar stressful conditions. That the pseudocoelom is an adaptation as a hydrostatic skeleton to allow them to survive better in the habitat that they live in. So there was evolutionary pressure to use it as a hydrostatic skeleton. And then on to developmental modes. We've already looked a little bit at this. I want to look at this blastula and give you a few more vocab words to use with it. So we said that all animals, it's a derived characteristic of animals that they have this standard development pattern. A diploid zygote, it's basically a fertilized egg, goes through cleavage. This is a lot of mitosis where the cells stay stuck together, 2 cells, 4 cells, 8 cells, and so on. And they ultimately develop into a blastula. This blastula is a hollow ball. If I peek inside, I can see there's a space. This space is called the blastocele. Cele just means space. Blastocele is not a coelom. The blastocele is just a function of the fact that this is a hollow ball. It's like inside of a tennis ball, there's a hollow space. It's just there because the ball is a ball. It has a space inside. When this blastula, we look inside of it, this blastocele, I want you to take a note that the blastocele does not become anything in the final embryo. It's just a function of having a hollow ball of cells. When these cells go through gastrulation and they infold and they form this archenteron right here, this primitive gut, archenteron. This is the formation of tissues, the germ layers. We develop here the ectoderm and the endoderm. This is a picture of a diploblastic organism developing. Notice how the blastocele is getting squished away as the endoderm folds in. The archenteron, the developing gut, is the space that's being created as the endoderm folds in. So it's this space inside. When we talk about this process, this can happen in 2 different ways. We can fold in at this folding in moment and form the mouth, or we can fold in at this folding in moment and form the anus. We're going to create the digestive tract. The blastopore is the little place where this invagination, this gastrulation, starts to occur. Depending on whether the blastopore becomes the mouth or the anus, we classify these organisms as protostomes or deuterostomes. Proto means first. Stome means mouth. Mouth. Deutero means second. Either you form your mouth first or you form your anus first? Anus first, mouth second. You'll be shocked to discover that humans are deuterostomes, and it may explain a lot in your life if you know that everyone that you have ever met actually started out and they were, just for a little bit of development, just an anus. Protostome is the ancestral trait of most bilateral organisms. That means with this group, as the coelom develops, so within the bilateria, this true coelom develops, and we have this purple marked off clade where all these organisms have coeloms. Protostome development also evolves here. So we'll put TC and P, or pro. Protostomes evolve with true coeloms. These are mouth first in gastrulation. All of this purple clade group, all of the organisms that had the ancestor with the true coelom, have an ancestor with protostome development. Now I want to show you some other things, because protostome doesn't just mean that the mouth develops first. Some other things go along with that. So when you develop as a protostome, let's say you're a nematode worm, when you develop as a protostome and you go through that first bit of cleavage, the pattern of cleavage, the pattern of development is spiral, which means that as the cells go through cleavage and mitosis, they create a pattern sort of like this if you're looking at the top down. Or if you notice here, there's no place, if I wanted to cut this embryo in half somewhere, if I wanted to try to cut it maybe and split it into two, there's no place here where I could cut it and not pop one of the cells. This is spiral development. Determinate means that these cells very early on have fates, they have roles, their fate is determined. That sounds kind of magical, but that's literally what we say. The cell fates are determined very early. They already have defined roles in their body. Protostomes have spiral cleavage, and their development is determinate. Cells' fates are determined early on. As gastrulation proceeds, the mesoderm, remember these animals are within the bilateria, they have mesoderm. Their mesoderm forms these little packets here in between the endo and the ectoderm. Here, they basically form as little packets, and the coelom is the space in the forming mesoderm. And the blastopore, as it develops, becomes the mouth. Well, mouth first, anus second. And this ultimately becomes the mouth to anus pathway, the digestive pathway of this organism. And if you look at a kind of a modern nematode, this is a pretty good drawing actually. Here's his mouth. It takes food in, it passes through his digestive tract, and there's his anus. It's not that far off of this organism. Deuterostomes, things like you and starfish, and deuterostome development occurs all over the animal tree. Protostome is ancestral, but deuterostomes are found within a lot of different groups. We're going to just show you two deuterostomes. The group that we belong to, our phylum, and the phylum that starfish belong to, but it is found across the tree of Animalia. In some of those taxa that have evolved deuterostomes, in all of the taxa that have evolved deuterostomes, we see a pattern of development that is radial, and this is cleavage. So as we go from a zygote to a 2-cell embryo, to a 4-cell embryo, to an 8-cell embryo, look at this. I could here take a very thin knife or a piece of hair and cut my embryo in half, and I could get even slices of it. This pattern of cleavage, or the pattern of mitosis during early development, is radial. It can make equal slices. It also is indeterminate development. These cells very early on do not have their complete fates developed. That means that they can take on the role of other cells. We'll talk a little bit about this during development. What that means practically is that I really can, at some point very early on, somewhere around here, cut this embryo in half, and this side can be separated from this side, and I can get identical twins. Each one of them would then contain enough information, and the cell's fates in the final organism haven't been determined. They can take over the roles of some of the missing cells, and they can each develop into a complete organism. Deuterostome can have identical twins. Protostomes cannot, because they have determinate spiral cleavage. Now, in deuterostomes as well, we have a different pattern of mesoderm development. As this gastrulation occurs, and we have the endo, the inside, and the ecto, the outside, the mesoderm doesn't form in these little packets. The mesoderm originally appears at the back of the archenteron, the back of the developing gut, and then it breaks off and it fills in this space in between the endo and the ectoderm. And then it creates the coelom here. These are the coela. And this is a cross section of this animal. It's not two sides. These are connected to each other around the back. It's just not clear. And finally, in deuterostomes, the anus develops first, the mouth develops second, the blastopore becomes the anus. Now I want you to compare and contrast these. I am not going to ask you on an exam, does deuterostome mean anus or mouth first? Everyone's got that. You know that this means anus first, because it's kind of funny and memorable. I'm going to ask you if deuterostomes have radial or spiral cleavage. I'm going to ask you where the mesoderms develop in deuterostomes, and ask you to remember and maybe compare and contrast deuterostome and protostome development. So I'll leave you with a question. You're swimming, standard Gen Bio 2 question. You're swimming, you find an animal that has spiral development, and it develops its mesoderm in packets. What additional characteristics do you hypothesize this animal to possess? Is it multicellular? Does the blastopore become the anus or the mouth? And does it have radial symmetry? One or many of these could be the answer. And I'll see you in the next video. I thought she was going to tweet for us, but I'll see you in the next video. All right. Bye-bye.